So I wanted to finish the lecture that we started on Friday by just doing in Model 5 questions 33 and 36, which I don't think we did justice to. And then I'm going to go back to Model 3 and look at gametogenesis and fertilization because we didn't cover that. Okay, so let's look first at Model 5, question 33. Model 5 is a condensed version of Meiosis 1. Notice the two possible arrangements of chromosome in late prophase 1. Considering what you know about DNA replication and meiosis, is either arrangement equally likely during the formation of tetrads in late prophase 1? So what we're saying is, what they're asking you is, these are the two possible arrangements, right? And remember, this is independent assortment. They can, if this is the metaphase plate right here, which is the line in the middle of metaphase, they can either line up with a large white and a small black on one side and a large black and a small white on the other, or the two large black, small black on one side and a large white and a small white on the other side. And they're saying, is it either of these two, is any one more likely than the other? And it's no, it's not. It's completely a random event, and that's the nature of independent assortment. Okay. Then it says, let's go to question 36. As a group, choose one set of daughter cells in late telophase 1 from model 5. Imagine that those cells now undergo meiosis 2. Draw at least 4 resulting haploid cells that could result. Okay, so let's just take a look at this. So let's just first understand, if this is how they line up and recombination occurs, it could go, these are two possible events that could happen. So let's take a look here. One cell, this cell, contains the large white and the small black, large white and the small black. And this cell contains the large black and the small white, large black and the small, and the small white. What's the difference? The reason why they don't look the same is because there was crossing over that occurred during late prophase, right? And so you have parts of the black on parts of the white, and parts of the white on parts of the black. And therefore you get some crossing over, which increases the variability, right? So that's why they don't look. Now, what's the difference between these two and these two? That's just different recombination. You can do it yourself, but you'll see is these are just different recombination events that are taking place here as opposed to here. So what they're asking then is, if you take these two, right? It says, take one set of daughter cells from late telophase 1. Let's take these two. Imagine that they undergo meiosis 2. What is that going to look like? So what I did was I drew that out on the next page because I really felt you needed to see that drawn out. So give me 1 second and let me get rid of these lines here. Clear my drawings. There we go. Oh, okay. So now I'm just going to back up really quickly. This is the original cell in late prophase 1. If no recombination had occurred, like, this is what it would have looked like. These two cells, it would have been all, now it's red has been replaced, red replaces white. So now it's all red, a big red, small black, big black, small red, okay? That's what it looks like without recombination. But in the figure 5, we have recombination. So this is this. So this is that top pair of cells in meiosis at the end of meiosis 1 in figure 5. So what is going to happen now in meiosis 2? Well, the first thing that happens is they have to line up on the metaphase plate. Notice that they don't line up on the metaphase plate with the two homologous chromosomes, right? They line up just like in mitosis, they're all lined up in one line, like this. So one of the cells, this cell, looks like this, and this cell looks like this. And here I put in the dotted line for the metaphase plate. So let's just take a look at this. What's going to happen? What is this going to look like after meiosis 2? So what's going to look like is this guy here is going to be that there. So it's going to be an all red one, big one, and then this guy here, a small black one. And then you're going to have here a black, red, black on this cell, and then a black red one on here, right? That's going to be the final product. And this guy, you can do it for yourself, is going to look like that. So that is the answer to question number 36. If you took the top pair of cells, um, if you took these guys here, that's what it would look like. So, however, not necessarily, because why? Because it all depends on how these guys line up on the metaphase plate. Right now, we have the red is on this side, and the black is on, and the black, red, black is on this side. But if you rotate this, right, 180 degrees, all of a sudden what will happen is the black, red, black will be on this side, and the all red will be on this side. And now when you divide these two cells, when you break the sister chromatids and the two chromosomes move into, you're not going to get this pattern. And that's what I put here. What I'd like you to do is switch these around and maybe switch one of these around and then see what the products are. And you'll see that the products are different. So how these sister chromatids line up here is going to affect the final product over here. Okay, so that is model 3. Now let's go to model 3. So model 3, we're looking at, it's called gametogenesis and fertilization. Gametogenesis is the formation of gametes, right? The sperm and the egg, and fertilization is the formation of the zygote, right? The fertilized egg. So let's just say here is at the end of meiosis 1, right? You have the spermatocyte, then that goes into, at the end of meiosis 2, you have the spermatids, and then you have the mature sperm. And here you have the secondary oocyte. This is the secondary spermatocyte, and that goes into the another secondary oocyte plus the polar body, and finally then you get the zygote. So let's answer the questions. According to Model 3, what is the name given to the cells produced at the end of meiosis 1 in males? So at the end of meiosis 1 in males, we call it a secondary spermatocyte. At the beginning of meiosis 1, it's called a primary oocyte, but now it's a secondary spermatocyte. Sorry, spermatocyte, not oocyte. What is the name to the cells produced at the end of meiosis 1 in females? And the same thing, it's a secondary oocyte. At the end of meiosis 2 in males, you have what is that called? It's called a spermatid, right? What do these cells from the previous question eventually become? They eventually mature into sperm. Before fertilization, what happens to the secondary oocyte? Before fertilization, what happens to the secondary oocyte? Well, what happens to it, it undergoes a division, right? And you end up with the secondary oocyte here and what's called a polar body. And what that means is that the polar body is just the DNA itself that's not in here, right? So it's the rest of the DNA that comes from here is now in here, but with no organelles, right? It's just the DNA itself, okay? During fertilization, which two cells come together? Be specific in your answer. And that would be a secondary oocyte and a sperm, a mature sperm. And that's happening right here. During meiosis 2, this is number 22, the secondary oocyte divides unevenly with one cell, the ovum, receiving half of the chromosome and nearly all of the cytoplasmic organelles, while the other cell, the polar body, is much smaller and eventually degenerates. With your group, propose an explanation to explain why the secondary oocyte divides in this way. And I am curious to hear what you guys said, but what we think the reason for this is that there's a huge amount of resources invested in making all the organelles and all the biochemicals in the cytoplasm of the secondary oocyte. And if you made 4 of them, that would be a huge amount of stuff here that you'd have to make and invest. What the body chooses to do is it puts all its eggs in one basket. It actually produces only one viable secondary oocyte and then 3 polar bodies. You don't see the other 2 polar bodies in this, but we'll see that next week. The sperm doesn't need to do that because basically the sperm is basically a polar body. I mean, the sperm is just basically DNA. So it doesn't have a lot of resources, so there's no problem in making 2 copies of it. So that's the gist of it for that question. Okay, I think that answers that. Feel free to send me more questions about this if you'd like.